Now for something completely different. I'm going to be talking about HS rats, and we're trying to put up this slide each time just to remind people that this is 8 inbred strains mixed for 100 generations. That's why we had birthday cake at lunch, right? And they're maintained by Leah Solberg Woods, who's over here in the corner, who's not at the meeting today. I don't actually know if Leah's online or not, but so Gattaca was a dystopian science fiction movie in the late 80s that some of you are familiar with, where they used pre-implantation selection to identify people with high fitness values according to their own standards. And those people became the class of what were called the valids, and they were sort of an upper class. And then the other people that hadn't been through this genetic selection technique were known as the invalids, and they were sort of an underclass because they were meant to be genetically inferior. So that's the concept of, is that okay? Okay, excellent. Yeah, all right. So people who haven't watched it, you might look at it. I, of course, have nothing to do with the movie, but we are talking about RATTACA today, which is very similar to Gattaca, as you'll see from the movie poster here. But the difference is that in RATTACA, what we're going to do is we're going to use selection that we do when the animals are young. We're going to genotype them. We're going to predict phenotypes in those animals, and then we're going to distribute animals with predicted phenotypes to other scientists. And I'm going to tell you that that's like the punchline of the whole talk. I'm now going to tell you about that in a little more detail, but just remember Gattaca, RATTACA, and I have to credit Paul Meyer, who came up with the RATTACA idea, I think just last year or whenever the IBANKS meeting was here in Memphis. Okay, so this is a new, exciting, remember, you should wake up even though it's 4:02 in the afternoon, depending on your time zone. This is new and exciting, a new paradigm for examining genetic correlations in outbred rats. And fundamentally, it works because for the last 10 years, lots and lots of our colleagues have been busy phenotyping huge numbers of HS rats. So at this point, we have a database of about 15,000 animals that have been phenotyped or genotyped. And that enormous database allows us to predict in future animals what their phenotypes might be like. Now, I should say, just to clarify, they haven't all been phenotyped for the same thing. They've been phenotyped for lots of different things. But as you saw, for instance, in Apurva's talk, we have some phenotypes that many different groups have looked at so that we get into the high thousands of animals that have been phenotyped. So we have this huge database. And then we can use very, very standard, and I want to emphasize, we have not invented anything here. We're using polygenic scoring, also known as polygenic risk scores, also known as polygenic indices in the animal breeding field, known as genomic estimated breeding values. Lots of different acronyms for essentially the same idea. We're going to sum up the effect of lots of different SNPs without regard for which ones are true and false positives in order to get an estimated value, estimated phenotypic value of a new animal where we only know the genotype. Okay, and this is just building on very standard. This is something that's been going on for a long time in a lot of different fields, both agriculture genetics and more recently in human genetics. So why would somebody want RATTACA rats, right? Like other than the fact that they have kind of a catchy name and it's sort of funny to talk about for 5 minutes, why would anybody want this? So lots of neuroscientists actually have models set up, and mostly not geneticists, have models set up where they screen cohorts of animals. Sometimes they're outbred animals, sometimes they're inbred animals, but they screen them to identify cohorts of high and low animals. And then they look at other things in those high and low animals, some secondary trait, and it's often something that they think maybe is causally related to the first trait. So an example, we worked with Terry Robinson at University of Michigan. They ordered huge numbers of Sprague-Dawley rats. They would phenotype these rats for something called Pavlovian conditioned approach, which it doesn't really matter. It's some behavior, who cares, right? But they would screen them for this. They would get a high, an intermediate, and a low group. And then they would give them to all of their friends who would measure all kinds of things about slice electrophysiology, gene expression, other behaviors, et cetera, because they were interested in these high and low groups. They were interested in individual differences, which is a terrific thing to be interested in, totally supportive. Obviously, everybody in this room is happy that people want to study individual differences. But we'd like to do it in a way that's unconfounded. So one of the problems can be that the measurement of the first thing might confound the second thing. And so, for example, we worked with a group that was interested in cocaine self-administration. So they would have these animals hooked up to an IV line. They're administering cocaine for a month and a half, very rigorous thing. Some of them will take huge amounts of cocaine. Some of them will take very small amounts of cocaine. That seems important, right? Like maybe you should study that. And so they did all kinds of things, but the animals from the high group and the low group, for instance, single cell RNA-seq and ATAC-seq, really exorbitantly expensive things to characterize the differences behind the high and the low group. And a reviewer reasonably asks them, are the differences that you're observing between the high and the low group the differences that drove them to take a lot of cocaine or not very much cocaine? That would be interesting. Or are they the result of exposure to a lot of cocaine or not very much cocaine, which could also be interesting. But when you tell me there's a difference between the high and the low group, I don't actually know what that difference represents, right? So what we're now working with that group to do is to provide them with animals that are genetically predicted to take a lot of cocaine or very little cocaine based on that GWAS that we've done for that very trait. But the animals, of course, have never seen any cocaine. They're completely naive animals. So now we can look at the genetic differences, the gene expression differences, the whatever differences that are associated with the genetic predisposition to take a lot of cocaine or not a lot of cocaine without having to be confounded with the cocaine. Same thing could apply for Pavlovian conditioned approach or any other number of things that people might want to sort the animals out for. And in addition, oftentimes people are using commercially available populations like Sprague dollies. It turns out actually the group that was doing Pavlovian conditioned approach, they would order the Sprague dollies and their vivarium would just get Sprague dollies from whatever vendor had them that week. And so sometimes they were getting them from Harlan, but other times they were getting them from Charles River, and sometimes they came from Mississippi, but other times they came from Toledo, Ohio, et cetera. So there was this huge population structure that was superimposed on their study. And a lot of the correlations that they saw were probably correlations driven by that population structure and not by any really mechanistic relationship between the first and the second thing being measured. And because it's not a homogeneous population, and because a lot of the groups that ask these questions don't think about genetics in the way that some of the people in this room think about genetics, that's sort of an unanticipated problem. Other approaches to this problem that many of us are familiar with is people that will grab two inbred strains, and the inbred strains might be different for phenotype A, and then they would measure phenotype B, and then they would make the case that phenotype B and phenotype A are biologically related to one another. But of course, you have albino rats, you have non-albino rats. Some of them are big, some of them are little. That doesn't mean that albino causes bigness or littleness, right? It's obviously a weak way to do it. So RATTACA, I think, can address these sorts of problems because we can give animals behavior, we can give people behaviorally naive animals that are pulled from one uniform population, and we use the genetic prediction that we've learned from all of these GWAS studies we've done in order to do that. So what do you need to do RATTACA? You need obviously to have the big database that I've already alluded to, where we have genotype and phenotype data. We wouldn't be able to make predictions about phenotypes we haven't studied previously, right? It's limited to the things that we have data about. We need to have the ability to collect tissue and genotype these animals at a relatively young age, since most people wouldn't want to get elderly rats or dead rats. They want to have young rats. It's the population that they're used to studying. And then we also need to have a pretty big population of these animals to select from, because if you were only making a population big enough to supply the orders for the RATTACA rats, then kind of you'd give them the upper half and the lower half and say, this is your high group and this is your low group. But what we'd really like to do is give them, say, the 5% highest and the 5% lowest. And that means that we have to have a big population to select out of. And it turns out because Leah's lab, who maintains the HS rat colony, and our lab that currently has a satellite colony of HS rats, because we're producing lots of those rats to give out to people that are doing genome-wide association studies and other kinds of studies, there are a lot of rats around, and therefore we can do this genomic selection and we can send people animals that are from the very edges of the continuum. And if we didn't have sort of an ongoing project to produce these HS rats, that wouldn't be a practical thing to do. So very briefly, you can imagine how this would work. When we're weaning the animals, which is at 3 weeks, we take an ear punch from the animals. We can then make that into extract DNA, make that into a library in about a week. It takes about another week to call the genotypes, takes about another week to make the predictions. So by the time 7 weeks have passed, they're 49 days old, kind of a young adult animal, we have predictions, and we can then send people out these animals. And what we're typically finding is that people want 12 or fewer from the high and the low group, and we're pulling them from a population of about 500. So that means we can give them the kind of the highest highest and 5 lowest percent of the animals. And we also are finding that people are requesting different kinds of traits. Obviously, if everybody wanted the same trait, then they would be competing with each other to get the high and the lows. But because they want different traits that are largely orthogonal, it works out. So here's an example of when everything is perfect. Over on the left side, on your left side there, we have along the y-axis our predicted values. Along the x-axis, we have the observed values. So what we're doing here is training on 70% of the data that we have and then predicting into the remaining 30%. And you can see that for a trait like body weight, where the heritability is quite high, 0.47, you can see along the top axis that the animals that we select as high and low are almost non-overlapping. They're really quite divergent from one another. And on the panel closer to me, the last one again shows mass, body mass, and you see they're extremely different from each other. So that's a p-value of like 10 to the minus 8 or something. The groups are extremely different. As the heritability is lower, and you see the heritability is written across the bottom, as the heritability is lower, obviously our prediction isn't as good, but it's still quite good. And even the worst group with a heritability of 0.13, which in our experience is pretty low, that's still a p-value of 10 to the minus 3 or something different group. So that we're able to make them pretty different. The other piece of the story that's critical to know about is it matters what the sample size is. So on the left side, I'm showing you 4 different heritabilities, the same 4 traits as before. But now I'm showing you, depending on how many animals we allow ourselves to use for the trainings, we kind of artificially limited the sample size and then resampled, I think, 10 times for each of those points. Obviously, the more animals we have, the better our prediction gets. But for most of the studies that we have 500 or more, and you'll see that by 500, even at the lowest heritability, we're already getting pretty good prediction. And then the panel closer to me shows you that you really don't need many SNPs to do this. So by the time we get to about 10,000 or 100,000 SNPs, we're already saturated. So although we can get millions of SNPs, we actually LD prune them. You don't need nearly that many. What can we predict? Here's sort of a smorgasbord of some of the different traits that we can predict, but the real answer is just email us and ask us for what you want, and we'll tell you what we can do. But you see lots of drug self-administration traits towards the bottom. Towards the top, we have things like locomotor activity that Apurva told you about, body weight, et cetera, Pavlovian conditioned approach that I alluded to earlier. We can predict other things too. So we can predict gene expression. So that group that had done the study with the single cell RNA-seq, and they wanted to know whether that was a consequence of the genotype or the exposure to cocaine, we actually predicted what we thought gene expression would have been in the high group and the low group. And then they were able to compare predicted gene expression across many genes to observe gene expression to see that, in fact, some of the gene expression differences were driven by the genotype of the animals, basically by the EQTLs is what we're using for that prediction. So that's another use case kind of. Another use case is that for a significant minority of genes, we have pretty good heritability of gene expression. So about 4,000 where the heritability is 10% and about 1,000 where it's 30%. So for those genes, if you happen to be interested in that gene, we could give you animals that are predicted to be differentially expressing those genes, kind of like a knockdown or an overexpressor. And then there are also a few hundred loss of function alleles that are banging around in the population. So for that subset of genes, we could give you animals that are like essentially like knockouts and wild types. So those are some other things we could do. And then just briefly, I think I have a minute or two. Is that 1 minute? Okay, great. So here are some use cases. This is Marcita Kalupi, who was interested in these high and low cocaine animals. And she wanted to look at something called spontaneous, I think I wrote it here because I knew I wouldn't remember, spontaneous inhibitory postsynaptic currents in slices of amygdala. And so she got animals predicted to be high or predicted to be low for cocaine. They had previously seen that this trait was different in the animals that actually had taken a lot of cocaine or not a lot of cocaine. And it turns out even in the genetically predicted animals, you see differences. And there's a significant interaction down there in the lower right. Another example is Greg Collins, who confusingly is at the University of Texas Health Sciences Center, which you also abbreviate UTHSC, but a different UTHSC. And he's interested in this drug, which is like a rave kind of a drug. I think it's in bath salts, MDPV. It turns out he had Sprague Dawley animals that took a lot of cocaine or very little cocaine, and then they took different amounts of this drug, but it wasn't clear whether or not that was like a causal correlation or due to the exposure to the cocaine that was driving the thing. So he was able to look at these animals and also saw a difference in these animals. So I'm out of time, but let me just thank everybody who's contributed, which is all the people that are generating the phenotype and the genotype data, and particularly to Ben Johnson, Tiago, and Bobby, who've worked a lot recently on some of the finer points. Okay.